This is Group 302, acetylcholine signaling, Lucas O'Brien and Ethan Wu. Acetylcholine signaling is a pathway that uses ligand-gated ion channels and utilizes acetylcholine, an endogenous neurotransmitter, to help govern voluntary muscle movements and many involuntary physiological functions like cardiac contractions and blood pressure. In this video, we will be explaining the role of acetylcholine signaling and doing a deep dive on acetylcholine signaling. Enjoy. Ligand-gated ion channels are usually used to transmit signals across the nervous system, as they cause rapid changes in electrochemical gradients, which allow nerve cells to communicate with each other. The polar hydrophilic signaling molecule binds to the extracellular binding site in the membrane-embedded protein receptor. The receptor becomes active and the embedded protein opens, allowing the flow of ions into the cell, rapidly changing the concentration gradient, which triggers a response in the cell. This response is short-lived due to when the ligand leaves the receptor, the embedded protein closes, and the flow of the ions in the response are terminated. This results in a rapid and short-lived response. There are a lot of advantages and disadvantages of ligand-gated ion channels. For example, the major advantages are 1. They produce quick and fast responses. Additionally, acetylcholine receptors have high specificity, meaning that only the acetylcholine signaling molecule and certain antagonists and agonists can bind to the extracellular receptors in the embedded protein. The acetylcholine signaling pathway consists of acetylcholine, the primary signaling molecule, or otherwise known as a ligand, and the acetylcholine receptor, which in this case is called the nicotinic acetylcholine receptor. When acetylcholine is released, it binds to its respective receptor. The acetylcholine receptor is a protein embedded in the plasma membrane, and it acts as a ligand-gated ion channel, which allows the passage of sodium and potassium ions. The acetylcholine signaling pathway begins with the release of acetylcholine, and then it binds to the acetylcholine receptor, and the sodium channel opens. Sodium ions then come flooding into the cell, called a large influx, while some potassium ions exit the cell, which result in an overall net movement of ions into the cell. This causes a rapid change in concentration gradient, which results in a change of events that leads to intracellular signaling, which results in a muscle contraction. Once the intended response is complete and a muscle contraction has taken place, the termination can begin. In order to terminate the signal pathway and relax a muscle cell, the necessary enzyme, which in this case is acetylcholine esterase, breaks down acetylcholine. The breakdown of acetylcholine removes the possibility of further receptor activation. Without a signaling molecule to produce a response, the sodium channel will change configuration and close. A sodium-potassium pump will then actively transport sodium ions into the extracellular space and potassium ions back into the intracellular space. This results in the concentration gradient reverting to its previous state so the pathway can be reused and the once contracted muscle is now in a relaxed state. Nicotine is a type of agonist for the acetylcholine pathway, meaning it increases activity in the synaptic cleft, where the acetylcholine receptors are located. Nicotine is exogenous and comes primarily from tobacco. When nicotine binds and activates the receptors, the response is longer lived than in acetylcholine, as nicotine is not broken down as quickly by acetylcholine esterase. This leads to increased stimulation in the brain, increasing neurotransmitter release, one being dopamine. Over time, a repeated use of nicotine products leads to receptor desensitization, which in turn causes tolerance and a dependence. This explains why nicotine products can be so addicting. Carrera is an antagonist for the acetylcholine pathway, meaning that it inhibits the activity in the synaptic cleft. Carrera is exogenous and comes from a variety of plants, one being strychnine. When binding to receptors in the neuromuscular junction, Carrera blocks and prevents acetylcholine from binding, which in turn prevents the flow of ions and a change in concentration gradient, which results in the prevention of nerve transmission impulses to muscular contraction. Therefore, the response is inhibited and results in muscle relaxation or eventual paralysis. This was our video on the acetylcholine signaling pathway by Lucas O'Brien and Ethan E. Thank you for watching.